I would say grew up in a part of LA, but it wasn't Bel Air, was it? Not Bel Air, not Rodeo Drive. At any rate, and then he and his brother Russell will come into this too. But he's not the, I guess I'm going to three people. But as you all know, they are like a force of nature through here. And it's sort of like the Campbell brothers. So I don't know, Adam could probably tell me. I don't know if this was probably through vertebrate biology we met these guys. And as you know, but the two of them, so I'm going to start with the two and I'll get to Dustin, don't worry, grew up with a keen interest in natural history. I would say mostly vertebrates, but even more so, a certain other kind of taxa that's not my expertise. Birds, as you guys know. So they were bird nerds of the first waters. All right, so put that on a pause there. Or that should give you pause, because like, did I read the title right? It's like, yep, you read the title right. Both of those guys were undergrads here. They worked in my lab. They had fellowships and were able to work on some data we had in garter snake work that needed working up. And so the other person I want to introduce is this gentleman sitting right in the front of the room, and this is Richard Hoyer. And Richard Hoyer is it's hard to define him. If I said he was an amateur natural historian, that doesn't cover it because he's published a lot in the literature. Is it not working to me? Yeah, okay. So he's published quite a few papers in the literature, so that doesn't count him as a naturalist, but he's not a practicing professional. He's a retired teacher, and so, but a natural historian of just immense talent. And that started, I guess, when you were an undergraduate here over with our friends across the sidewalk here, however many years ago. I'm not even going to hazard a guess there. But Richard is also a bird person. He's a master falconer, and he's trained a lot of falconer people. But why we're here is Richard is also really a natural historian who has spent enormous, tens of thousands of hours looking at herps, looking at snakes, but not looking at easy snakes, as he always likes to rid me, not garter snakes like I work in the tens of thousands in Canada. He works on the secretive, sneaky little guys that most people, even herpetologists, have not seen, especially what Dustin's going to talk about today. Okay, some of you know Mark Lepin, who also was able to, did his thesis work on rubber boas that was based on Richard's decades of mark recapture work. So then, of course, our favorite friend comes into then COVID, and then for us, the move to CRB. So COVID just shut us down in terms of Canada. It was closed down for 3 years. Then we moved to CRB. Why is that pertinent? Because there's no environmental chambers there. So it was like we were really high and dry. But at the same time, Richard had said, you know what? I'm getting up there, and I'm not a master experimental design and statistical analysis person. Do you have a student that could work? That I have this great data set, mark recapture set. You'll see 22 years worth of data that could analyze this. I said, oh boy, do I ever. And so these guys, sorry, Russell, you're out of the picture now. These guys, but Dustin, I knew was still in the area, even though he graduated. So we talked about that. I said, they're not birds. He goes, no problem. I'm your guy. All right. Take on the project. Yes. So this probably you'll see. I don't want to steal all of Dustin's thunder there. So this is a snake that's it is just, he'll talk about this, but I want to reinforce it right at the beginning. These are not like a garter snake or a gopher snake that all of you guys have found when you walk out there. I've lived here for 35 years. I have never seen one of these snakes just walking and doing my own thousands of hours of surveys. You just do not see these snakes. They are extremely secretive. You have to go out of your way to look at them or to find them and to work on them and so forth. So Richard has done that. And Dustin and his buddies, some of you folks have had that hit. So I also want to just end and say I recognize Dustin's family is here, and his partner Nicole, and So thank you for your support of him over the years. And then, is that me to man or?
No.
Oh, sorry. Oh, I moved. Oh, you're moving the camera? Sorry. Okay. All right. So without further ado, Dustin is going to talk about what he's been working on for his master's degree here. And that is growth, seasonality, and hatchling morphometrics of common sharp-tailed snake populations in Western Oregon, USA. All right, so let's switch off here. Transition.
Hello, everybody. Almost ready. Okay, as you have heard, I'll be talking about really life history characteristics of this little known snake, specifically the growth, seasonality, and hatchling morphometrics of this snake here in Western Oregon. So first, it's important to give like a background and the setup of what we're doing today, right? So we'll get in our background and introduction. So what is this snake, right? What is known about them and why are we studying them? And then we'll get into our findings, right? The population ecology and life history. That'll be seasonality, population size structure, growth, maturity, and hatchling characteristics. And at the end, we'll kind of bring it all together and talk about what it means and then where the avenues for future research. So obviously, as you can expect, we'll start with our background and introduction. So first, there's some terms I want to make sure that we all kind of have the same idea of. And the first one is population ecology, right? That's characteristics of populations and how they change over time and interact with their environment, right? And that includes birth and death rates, right? So how quick individuals are entering and leaving populations. And then the structure of the population, right? So the age and size distribution within a population. And this is affected by an animal's life history. And I've said that a couple times. So what is life history? And that's the timing and energy investment that animals put into reproduction, growth, and survival. And it's not really this uniform thing that all animals do it at the same rate, right? We have what we call slow and fast life history characteristics. And those are influenced by an animal's rate of growth, time to sexual maturity, and then its size at birth and hatching. Right? And so we have what I said, like the slow versus fast, really. And when we look at slow life history, we can think of delayed time to sexual maturity, right? So it takes them longer to start reproducing and add individuals to the environment. They typically have longer lifespan and they'll have slower growth, right? A good example there is a whale, right? Whales live a long time, they grow really slow, and they have offspring every once in a while. And the opposite of that is going to be fast life history, right? So that's quicker time to sexual maturity, rapid growth, and then shorter lifespans, right? A good example there is rats. Right? Rats reach sexual maturity as quick as they can and start reproducing as often, but they don't live as long. Right? And that can be influenced by environmental conditions. And so you might ask, why does life history even matter? Right? Why do we care about an animal's life history? And it's because it can inform about potential vulnerabilities of the animal, right? What is it susceptible to as far as environmental changes? And that information can inform management strategies, right? If we know how the animal lives and how it's investing its energy into its life, how do we manage it if it needs help? And then ultimately that can lead to changes in conservation status. An example here I give is nicely on the sharp-tailed snake, right? They're listed as endangered in Canada. And so in this paper, they looked at the life history parameters and see how that played into the conservation listing, right? And it's important because so that study came out of Vancouver Island, right? The north, that's that big, high northern limit of the population, right? And snakes are ectotherms, which really means that they require energy from the outside in order for them to do metabolic processes and be generally active, right? And so in that northern limit of their range, where we're going to probably have some different climate effects, you might not have indicative life history characteristics of what you would see throughout the rest of the range. So that brings us to Oregon, right? Because that's where we did our study. So when you think of Oregon, you might not think of like the most snake speciose place, but really we have like a decent diversity here. So within Oregon, we have 3 families. We have Viperidae, right? In that top left photo, you can see that rattlesnake hidden in the grass. We have Boidae, which is our boa. In that top right photo, there's me holding a rubber boa. And in the bottom right, which is Bob's favorite snake, a garter snake, right? Part of the family Colubridae. And so in Oregon, total, we have 17 species of snake. 15 of those are colubrids, right? And it's the most speciose family of snake, so it makes sense that it is our most speciose group. And I just wanted to show a little of the diversity of snakes we have in Oregon, right? So in that left photo, everyone's probably seen a gopher snake. They get pretty large, 5 or 6 feet. They have that tan and black kind of pattern on them. We have the garter snake, which I'd be surprised if anyone in here hasn't seen one. We have the feisty racer in the top right. In the bottom right, we have the mildly venomous ringneck snake. They have this beautiful under, like their ventral side, it's a nice yellow to orange to red. It's beautiful. And then it takes us to our star of the show, right here, the sharp-tailed snake, Contia tenuis, which would be under the family Colubridae. So they were first described in 1852 in Washington State, right? And they're a small semi-fossorial snake. And you might be asking, what does fossorial even mean? right? So these semi-fossorial animals are adapted to living under the surface, and not like deep underground, but just under the surface. A good example there is this image in the snow. It's kind of tracks from a vole, right? They won't be right on top of the snow running around. They're just under, or just under that surface level, and you see the tracks, right? And that's where they're adapted to living. And that is the same with the sharp tails. They'll be either under like leaf litter or gravel or what it may be. That's that nice top layer, but they're just under the surface. Right? And they're native to Western North America, right? So they call this home just as we do. And they're one of two species in the genus Contia, the other being Contia longicauda. And I want you to keep that in mind because we'll talk about it again in a second. And they are oviparous, which just means they lay eggs. And so what are the diagnostic characters, right? How do you know you're looking at a sharp-tailed snake? And one is that sharp terminal scale. I have a pointer, so I don't have to leave the camera range. So if you look right up here, on the left side, this is Contia tenuis, the sharp tail, and on the right is a garter snake, right? And you can see that even though you get like a bit of tapering to a point on that garter snake tail, it's not a really strong, distinct point like it is on that sharp tail snake, right? And there's been no formal study on the use of it, but there's some ideas. And some ideas are, one, it could be predator defense, right? Like using that tail to jab at predators to ward them off, or for prey acquisition, right? Using it kind of as an anchor to grab onto prey and pull them in, right? And then they have this black and white barring on their ventral scales. You can see that ladder-like pattern underneath. And that has like, it comes with a unique kind of behavior they have where they'll knot up in a ball like this. And there hasn't been too many formal studies on why they do it, but the current speculation is to mimic millipedes. Millipedes have toxins, and it's just a kind of way to be like, hey, you might not want to eat me, I might be dangerous. So they kind of do this weird nodding behavior to display their ventral side. And they're either going to be black and red or have that roll, like dullish red-brown coloration on the top. And then they're generally a small snake, being under 400 millimeters in total length. That's from the tip of the nose to the end of the tail, and that's under 16 inches. And then we need to know what to eat, right? We talked about that it's potential for prey acquisition in their for the tail use. So what do they eat? They seem to have a strong preference for slugs, and that's looked at in the morphology, right? Slug eaters typically have a larger and more recurved teeth than you would expect for their skull size, which they have. And there's also been slugs found in the gut content. And I think what's really interesting is they have this strong association with this genus Arion, which is an invasive European genus, which was introduced in the Pacific Northwest or first found in 1933. So they seem to be making use of this new, relatively new invasive resource, right? And so for where they live, right, you've seen this map before, right? They live down to the bottom of the Sierra Nevada mountains in the coastal range in California. And they reach all the way up to that southern tip, right, that northern population in southern Vancouver Island with some disjunct populations in Washington along the way. And then within that, right, we have their general geographic range. So where do they live within that, right? So they really like these forest edge or oak savannas and rocky outcrops. Really, these areas that have open space for a lot of sunshine to come through, but also have this nearby effect from trees or forests, right? And I wanted to include this image of a nearby site that's quite fruitful and very lucrative for these snakes. You can find them kind of everywhere. And then you can see it's next to some agricultural land, right? It's next to a Christmas tree farm. But you see a lot of these same characteristics in this site, right? So we have the southern facing slope, and this south slope allows it to get sunshine more throughout the day. And then you can see these kind of disjunct trees spread out throughout, right? And then as well as the rocky substrate, right? So even with like this human disturbance, they seem to be managing fine in that area. And because a lot of these things I just mentioned, they're generally a hard to find snake, as Bob has mentioned, right? That semi-fossorial nature is just kind of intuitive, right? It's harder to find something that's just underground than something above ground, right? And they're small body. So again, the same kind of thing. It's hard to find a really tiny snake, and you'll see an image soon how small they are, than it is to find a big snake. Again, no descript, really stark thing about them. And because of all that, they're not commonly encountered unless you're searching for them. I'm sure everyone here has kind of stumbled across a gopher snake or a garter snake and noticed it, but the odds of you running into one of these sharp tails is very low, right? You kind of have to actively be looking for them. And then also, we don't really know about their active seasons, when they're really out and about doing what they need to do to survive. And all that leads to them being generally poorly studied, right? And as I mentioned, they were described in 1850. And since 1852, they have 26 research publications today. So not much done on them over all of this time. And you would expect to see a lot of that research out of that bulk of their range, right? That California and Oregon. That's where you would think we get our information from. But surprisingly, you're getting it out of that northern population in Vancouver Island, right? And I mentioned life history characteristics and how that can play in, and how climate can play into that. So it's interesting to see that we get all of our information on the snake from that northern population. All right, so we have that extreme northern limit, and we're in Oregon, so it's important to compare the two, right? And how the climate shakes out between the two. So Vancouver Island has what is called a maritime climate, And that's categorized with high annual rain and cooler mean temperatures, right? And again, that far northern limit of their range. And then we have Oregon with a Mediterranean climate, which has warmer mean temperatures and lower rainfall, right? With the hot and dry summers. I know we like to think of Oregon, especially with the last couple of days, having like a ton of rainfall, but really we have these longer spells of hot, dry weather. And again, we're in that middle of their range. So, right, with what we talked about, why study continental, right? We mentioned how there's not been much on them over all this time, and they're really hard to get a hold of and really hard to find. One of the reasons we want to look at them is just that, right? There's not a lot known, and it's important to know what we can about the animals that are here while we have them. And this lack of knowledge has led to them being listed as endangered and vulnerable in our state of Oregon. And that doesn't come from finding information that would require them to be listed, but it's more that we have no information on them and don't see them, so the assumption that they must have some conservation issue. And that the known population comes from that extreme northern limit, right? And these populations on the Vancouver Island are relatively small, and thus their study and sample size was a lot smaller than the one we'll work with, which we'll get to in a second. So in their 9-year study, they had just over 100 snakes encountered, which that brings us to something that Bob had already introduced, which is Richard Hoyer, right? So he provided us with a large mark and recapture data set. And what mark and recapture is, you collect a snake, you give some identifiable marker on it so you can realize the individual you're working with, you release it, so then when you recapture that snake, you can get continued information on one snake. And Richard Hoyer has a long time dedication to life history collection. And as he said, he's partnered with us before, because Mark, he worked with his data on rubber boas in order to get his thesis. So he's worked with us to get these things worked on and published. And so the data set we're working with is 24 years long, right? It has 29 total populations throughout it, with 1,815 individuals, with a grand total of 2,388 capture events, right? And so when we compare that to the 9-year data, 9-year long data set with just over 100 individuals, we're working with an order of magnitude larger than the previous studies. And just to talk a little bit about Richard, it's because he's very impressive, right? He is still in his 90s and actively goes out into the field. It was like a month and a half ago, I received an email from him that he went out and kind of was like, oh, here's what I found in the field today. So it is surprising. I would like to be able to do that if and when I reach that age to be out in the field doing this stuff. And I'm giving him some acknowledgement here, but he's also been formally acknowledged for his dedication. He received the Lifetime Award at the Snake Ecology Symposium, which was to represent over 50 years of dedicated fieldwork as a herpetologist, right? So he's been doing this for a long time. And not only has he been out there collecting this data, but I told you to keep the Contia longicauda in mind for a reason, right? Because it was in 2001 that Richard Hoyer kind of brought it to our attention that we might be looking at a different species within Contia tenuis. So there might not be just tenuis and we see another species. And that was all based on morphology, right? So he noticed the tail length between these individuals seemed to be a lot longer, right? And if you look here, and you also might think, how do you know what a snake tail is? So if you look at that top image, on the left side, you can see that, let me get my points. So you see right here, this is the cloaca or the vent of the animal. Really, everything after that is the tail. And when you look at these two, we see quite a difference in tail length, right? And that's something that Richard noticed, as well with the less stark ventral pattern on these individuals. So he made this publication about a problem new species, and then 9 years later, it was confirmed and described as its own species. So not only has he collected all this data and provided it to students to be able to work with, but he led to the description of a new species. And now that all brings us back to what we want to do with the data. So we wanted to examine the life history characteristics of the snake in Oregon, right? And then ultimately compare it with those life history characteristics found in that northern population on Vancouver Island. So the questions we really wanted to ask is, in what season do they have the highest observations during field sampling, right? So when are we seeing them the most? And then we wanted to know, what is the population size structure of males and females? So how, within a population, how are their sizes distributed? We wanted to know the growth rate of them in Oregon, right? Because that plays into that life history. We wanted to know the relationship between size and age. And then with that, the size and age then become sexually mature, right? Because time to sexual maturity also plays into that life history. And then we wanted to know if there was any sexual dimorphism in demographic or life history characteristics between hatchlings or adults. So that is our why we did it, right? And what we want to do. So now we'll get into not only our findings, but we need to talk about how we did what we did, right? Our methods and our methodology through this process. So I mentioned that we had 29 localities across Oregon, right? And you can see we have this big cluster of them here up near Corvallis, around where we live, and another cluster down in southern Oregon. And then that one, let me get my pointer. We get that one right here east of Mount Hood, that one population. So And fortunately, it's not this one uniform site, right? We had a nice elevation gain, those differences between the sites, with them ranging from 79 to 387 meters. So now, right, we have the sites and we know their suitable habitat. So now we have to find the snake, right? And finding the snake, the little snake that you can see in this image here, is not necessarily easy to find. It can be tricky. So what we do for sampling snakes generally is lay out artificial cover objects. And what that is what it sounds like. You take an object that's not typically there in the environment and you place it down and it works as cover for the animal. And in the image on the right, you can see a piece of corrugated metal. And in the middle, you see the carpet. There's other options like roof shingles, tile, really anything that you can sit out in the environment for them to hide under. And they can hide under it for multiple reasons, right? There can be nice thermoregulatory benefits from being under the metal. As the sun heats up the metal, they can hide under it, and right, they need energy from the environment. So they can get that from that metal and then do their daily activities. So you find your cover, you flip it over like in that second image with that folded carpet, and then hopefully you find your snake. And there's one other type of artificial cover I wanted to talk about because it's unique to the study, and that's the use of gravel piles, right? So a lot of artificial cover is, like I said, carpet or tin or tiles, but these are just what it sounds like. You take gravel and you pile it up and you rake through it to find them. And we'll talk about it again later, but it's unique to this research and seems to be an effective way to sample for them. So then we have to collect our morphometric data, right? So we want to measure our total length and our tail length, which how you do that is you put them up against some sort of measuring tool and you stretch them out and then you measure the total length, so the tip of the head to the end of the tail. And then you want to measure the tail length, right? So the vent into the end of the tail. And then if you subtract the tail from the total length, you can get your snout vent length, which is what we use for direct comparison with the Vancouver Island study. And then you want to measure the body mass. And the example in that image on the right is called a Pasola scale. You put the snake in a bag, weigh it, and then when you can take the snake out the bag, you have the weight of the bag, and then you can subtract that and get just the weight of the snake. And then we need to, right, it's a mark and recapture. We have to be able to identify the individuals recapturing. And so Richard had a unique way of identifying these individuals. Typically what you see is people will take clips of like the ventral scales of the snake, and you do it in a unique pattern. So it's unique to that individual and you know what you're working with. But that can be difficult for a snake that is very little, especially young ones. So Richard found that there was unique characteristics in the scale structure on their heads and was able to look at that along with other defining characteristics like scars or different patterns on the snake and be able to find it accurately to individual. And that was tested in lab and he was 100% accurate. This was a viable way to identify the snake. And here's an image of the scales and how they're laid out, right? We have our labials, we have our parietals. So these scales and the surrounding scales and looking how they're structured and how many there are was a way to find them to individual. And then we had to figure out how we know if a snake is sexually mature, right? And so one way is the presence of gametes, right? You can look for sperm in males or eggs in females, right? So you can like feel on the female's body and feel for the eggs and you know it's there. Males, it's a little more difficult because you have to look more on the cellular level, but it's possible. Or you can look for one expressing reproductive behaviors, right? So actively courting a female or a male, right? The other snake. So now we have all of our information and we have to manage this massive data set. And so again, this data set has 1,815 individuals with a grand total of 2,388 captures. It spans 24 years. It started in 1998 when I was only 2 years old. It's really interesting to work with a data set that was started being collected when you were only 2 years old. And per capture, we have so much information, right? So the date it was captured, the location, which individual it is, and then all the morphology, right? So the body mass, total length, snout vent length, tail length, and then any reproductive information. And now with the data set, the fun part, right, is the statistical analysis. So we used RStudio for all of our analysis. We really did a combination of two-tailed t-tests. All right, and that allowed us to compare the males and the females, right? If we wanted to find a difference between what males and females are doing or any characteristics, that allows that. And then we did a Tukey HSV post hoc test, which is done after an analysis of variance, right? That's what the ANOVA is. And that allows really for you to cross compare a bunch of different groups and find differences between them, right? So then we get into what we found, right? And so the structure for this results section, we'll talk about seasonality, population size structure, right? The male versus female. We'll get in our growth rate, both juvenile and seasonal growth rate. We'll talk about size at known age and the size and age of sexual maturity. And then we'll switch gears and talk a little bit about hatchling morphometrics. So for seasonality, we got information from the US National Weather Service, right? And we used multiple cities and locations across that map we had, right, where all the field sites are. So we get an idea of the climate that like averaged out what they're all experiencing. And then we created a histogram with the monthly observations. And all that is we took the date information we gathered, right? So the month, day, year, boiled it down to just the month, And then so over this 24-year span, what months had the most snakes? And so what we find is that you get these peak observations in the spring and fall, right? These seasons of more moderate temperature and rainfall. And I think it looks really nice here where you can see, right, where these two peak out is exactly where the black line, which is our temperature, and the gray line, which is our rainfall, right where they intersect, right? So we're seeing this high amount of observations in snakes relatively surface active right during these seasons. And we have our lowest observations in the opposites, right? Our more extreme seasons. So we have these dips in it all when we're at summer, which is high temperature, low rainfall, and in the winter, which is low temperature, high rainfall. Right? And then so we wanted to look at our population size structure. So we plotted our snout vent length against body mass, right? And divided for males and females. And we found is generally the females are heavier than the males. So when you look, kind of here's the mass on this side. These individuals that shoot way higher up later in life, those are the females, and it shows that they get heavier. There's also more variation in that max size, right? The right end of that graph kind of spreading out as much as it does. There's just a lot of variation in maximum size for females. And in this, we can kind of see these three observable cohorts, right? In this early one, right? There's really, it's almost just a line. It's really tight-knit group without a lot of variation. It's going to be our first year, right? There's not a lot of variety in the size of the first year. And then there's this little bit of a gap you can see in both of them, and it spreads out a little bit. It's not as tight. It's going to be our second year. So a little more variation, but not a ton. And then at the end, which is easier to see in the females, you have that third year and over, where you really get it jumping out and spreading out and being really disjunct, right? So you have a lot of variation in this female max size, while the males are much more confined to a generalized max size. And so I talked about how small they are, right? And so this is like a really little snake I had, and for size reference with my hand, it's a very small snake. And that one on the right is probably the largest snake concentration I've seen ever, right? And so this is like the far left of that graph, and that's the far right. And at some point, they have to kind of go from that size to that size, right? Which is why we wanted to know the growth rate, right? How fast are they doing it? So we wanted to figure out if there was a difference in growth rate based on that juvenile body size, right? So we split them into 4 size classes. That being hatchling, small juveniles, mid-sized juveniles, and large juveniles. And we did this because out of the Vancouver Island study, they gave the large juvenile growth rate, right? They couldn't get the other ones because they didn't have enough recaptures on the smaller juveniles, but they give the large. So I wanted to know, is there even a difference in growth rate based on juvenile size? And we found out there isn't, right? So across the board, no matter the size of the juvenile, they're growing at the same rate. But when we wanted to know how are they growing per season, right? We had data for every season, which isn't usually common for snake studies, right? People go out when they know that they're active, but we had it for every single season. So we wanted to know, how is it going across the board? What is the growth rate for each season and the difference between juvenile and an adult? And so what we find is that we have a really high juvenile growth rate in the summer, which if you recall the seasonality information, is kind of contradictory because we had really low observations during those months, yet they seem to be putting on a lot of weight and size during those months, right? And so they're growing at 13.46 millimeters per month. We found no difference in adult growth rate by season, so they grow kind of consistently across the board. But I do want to note that if we see in these adults in the winter, there are a handful that seem to be packing on quite a bit of mass in those winter months, right? And that brings us to why we might see a potential contrast in the seasonal activity and the growth rate, right? And that is because they're potentially facultatively nocturnal, right? We have that lowest activity in the summer, like I said, yet the highest growth rate in these juveniles. And so there's some anecdotal evidence and some evidence of captive individuals being more surface active during these summer months, right? And because of that, I think we should have more nocturnal focus on the studies for this species, right? Because we don't know exactly what they're getting up to in the summer, and they may be more surface active than we see in other months. And then that takes us to our size at known age. So we had to create a data set where we had individuals full where we know how old they are and what size they are. So for those, we set the hatching date to August 31st, which was the average date for eggs hatching in captivity. We used the last capture for analysis, and that's so you don't kind of just jumble it with useless information, right? The most important thing you get is what is the size of the individual and its age at the largest, right? And so for this, we compared 3 growth models, which are logistics, Gompertz, and von Bertalanffy, and we found that the logistic model fit the data the best. And you get this kind of interesting story out of this graph, right? Because you can see the angle of the curve, but when you look at the data points, it tells a little bit of a different story, right? So we see that some of these individuals have a much steeper incline than the model is predicting. So there's this potential overestimation of the size at known age, and that could be due to this huge bulk of data at the bottom kind of weighing it down. But either way, the numbers we get out of this may be a potential overestimation for size at known age, which really takes us into age of sexual maturity, right? So again, we kind of define that as the presence of gametes or exhibiting courtship, right? And so we took the smallest observed individual that fit those characteristics and plugged them into that model. And so the smallest male was 181 millimeters snout vent length, and the smallest female was 201 millimeters snout vent length. And so when we apply that to this model, we get those points there, and we figure out that the males reach sexual maturity at 2.4 years, and the females after 3.4 years, right? And as I mentioned with that potential overestimation, and we can see some of these points are shooting up sooner and hitting these levels before the model predicts, we might be seeing them reaching sexual maturity even sooner. And this is important and ties back to our life history because, right, we found the 2.4 years in males and 3.4 in females, but on the Vancouver Island study, they found them reaching sexual maturity in 5 to 6 years, right? So even with that overestimation, we're seeing this notable and noticeable difference in our time to sexual maturity, right? Which plays into our life history and thus management strategies, right? So generally the time to sexual maturity is faster in Oregon than we find in Vancouver Island. Right, so that is all of our stuff on growth and size and maturity, right? So I want to switch gears to hatching morphometrics because we got a really cool finding here. So all of the eggs and the hatchlings were collected and maintained by Rich Hoyer. He collected gravid females in the wild, let them lay eggs in captivity, and would release them. The eggs were hatched in captivity and they were held on a 12-hour light and 12-hour dark schedule, with the high temperature being 27.8 degrees and the low being 23.9. And then as soon as was possible after hatching, all the morphology was taken, right? The total length, tail length, relative tail length, and body mass. And in some ways, we found no differences, right? So total length, right? The length from the tip of the nose to the end of the tail, the relatively the same. Body mass, the same. And snout vent length was the same. But we did find differences in the relative tail length and the tail length, right? So males have longer tails and thus higher ratios of tail length to total body ratio, right? And that is a new finding and important because it gives us this non-invasive way to sex the snakes. So really the previous methods for sexing a snake is you can do what's called everting hemipenes, which right here, these are the male reproductive organs of the snake. And you kind of just like physically push them out and then you can identify it's a male, right? And the other option is probing, where you get like a metal stick and you feel for the inverse of this. So when that's inside the body, it's more like a pocket and you feel for the inside and then you know you have a male. The problem is, one, there's potential for harm of the snake, right? If you're pushing too hard, you might damage the snake. But also on a really small snake, like the one you saw in my hand, it's gonna be really difficult to kind of get those hemipenes out, right? So you don't have a viable way to sex them. And in the Vancouver Island study, they did not analyze their juveniles by sex because they didn't have a confident way to sex them pre-adulthood, right? And so also if you make mistakes with these, and these are not easy strategies, right? You kind of have to be trained and know what you're doing to evert hemipenes or know what you're feeling for with probing. But if you make a mistake, you can get faulty data, right? Finding a female as a male or male as a female and making this mistake, which will mess up the entire data, or not the entire data, but mess up your data collection, right? So then, right, that is everything we found, right? So then now what does it all mean, right? What are our conclusions and where are the avenues for future research? So a big comparison, right? We wanted to compare it to that northern population, right? We found that the Oregon growth rate is faster than the Vancouver Island. Where Vancouver Island, they found that they grow 19.5 millimeters a year. And if you recall, to what we found is that in the summer, the juveniles are growing 13.46 millimeters a month, right? And so when you scale all of the seasonal growth rate up, we're getting potential growth for 68.01 millimeters a year, right? And I know this high variance looks scary is because when you add up all the variance of the seasons together, it like, obviously you're not getting negative growth, but it shows the huge potential for growth in these individuals within the span of a year. And right, so we compare it to the same species in another range, right, with different climate effects. But then we want to know, how does it compare to similar species in different parts that are going to have more similar ecological or climate effects? So when we compare it to Steroria decay, right, decay snake, we see that they grow at their height at 16.2 millimeters a month, right? And we found at our height of growth, 13.46 millimeters a month, right? So we're seeing a similar growth rate to these ecological equivalents, right? This snake here is also a relatively small semi-fossorial slug-eating snake. So it's ecological equivalent in a different part of the country is showing similar to what we found. And I wanted to jump back to these artificial gravel piles. So artificial cover objects work best when they're suiting the needs of your target animal, right? You can think about a salamander probably doesn't want to be under a really hot piece of metal, right? It's hot, it's dry, it's not going to serve its needs, right? And these gravel piles create this almost underground or undersurface traversable environment that these animals can use. And it especially seems to be useful in these warmer months when they're harder to find compared to being under these really hot and dry cover work, right? And these also not only work as a way to sample for potentially any of these semi-fossorial snakes, right? And it includes sampling efforts, but it can be a conservation effort, right? If you need to create some kind of habitat or have suitable habitat for them in an area, these gravel piles may work as a way to give them more livable space. And I wanted to also talk about the diet again, right? Because we talked about how they were strongly associated with this invasive genus, but we see that they have a ton of growth in the summer, right? And what do we know about slag availability in the summer, right? We know that they have more nocturnal activity in the summer, which would track with the potential move to nocturnality in these guys. But these guys, again, are really small snakes. And I'm assuming you guys have seen slugs around here. Some of them get big, right? These guys are definitely not going to be eating on an adult banana slug, right? So they really need these smaller bodied slug or slugget, right? So is their diet flexible, or are they really locked in on these slugs, right? On only slugs. So I think a full diet analysis could help fill in the gaps in our knowledge and kind of answer these questions on maybe what they're feeding on in the summer. And then it wouldn't be modern day if you don't talk about potential threats to an animal, right? So there's the agricultural use of molluscicide. Molluscicide being toxins targeted at mollusks. In this case, we're really looking at slugs, right? Because slugs for agricultural are seen as a pest, but it's their primary food source, right? So if we're either removing the slugs or adding toxins into their food, right, this can be a threat to them and potentially harm populations. And we look at climate change, right? This map here shows worst case climate change scenario, how their range changes. Now, fortunately, we see that their range, they gain more than they would lose, but that doesn't tell the whole story of climate change, right? We have more extreme weather events. We have these longer, hot, dry spells that could also influence their food availability and various things that can go on in their life. And then it's important to think about fire, right? We get regular, large-scale fires here in these summers, and we don't know their ability to withstand and bounce back from a fire. So then that brings us to what do we get from all this, right? What are the general take homes, right? We got to work with an incredibly large data set, not only for this snake, but for similar ecological snakes, right? This 24 years and nearly 2,000 individuals is unseen for these difficult to study snakes. So that's one big part of this. And that gives us information from the center of their range, right? The previous data came from that extreme northern limit, right? And a unique climate for what a snake has to deal with. So this gives us information in the center of their range, the bulk of it, what they're getting up to. And then, so we found this difference in age to sexual maturity, right? Even with that potential underestimation, but generally we're seeing these much more faster life history characteristics in Oregon compared to Vancouver Island. And then I think one important thing to bring up is I think there needs to be more research done in California, right? When we look at their range map, that almost like 75% of it or whatever is in California, but almost all of our information comes from the northern ranges. And if we're seeing a difference in Vancouver Island and Oregon, it'd be interesting to see the difference in these life history characteristics in the other parts of their range, right? And also, I want to get back to that nocturnal activity. I think their activity in the summer, potentially becoming surface active, as opposed to staying under the cover, I mean, semi-fossorial, having more surface activity in the summer, is something we should learn more about. So with that, I want to get to my acknowledgements. So first, earlier I gave Richard Hoyer a more formal acknowledgement about the things he's done and his accolades, but I wanted to give a more personal acknowledgement for him, right? So not only, again, he provided the data for me to do this in the first place, but he's been incredibly helpful through the entire process, right? All it would take is a quick email to him, and he needed to be willing to take me out to the field, provide me with more information, clarify things in the data collection, right? He provided all this. And he would also, at the same time, invite me in his home to talk about these things and work through them. And he would always make sure, anytime before I left, that I got a cookie. And if you know anything about me, you know it's very important to give me a cookie. I want to give a thank you to the office staff. I think not only has like the end of my thesis been very accelerated, they've been very helpful in getting make sure we get all these deadlines and get everything set up. But I think just generally, they help us with any question and anything we need. And it'd be hard to do what we do here as grad students without the amazing support of them. And also just like moral support is a big part, right? They support us morally and make us all feel good, which is a nice part about the office staff. So thank you. I want to give a thank you to my committee, which similar to the office staff, as I said, this whole end of my thesis has been very accelerated. And they have been nothing but responsive and helpful to the entire process, right? It took one email with a set of dates to get a defense set, and it was done, which isn't a common scenario. It's not easy to get 5 people with the same schedule lined up to be in the same room for 3 hours, but they did it, right? We made it work, and I appreciate them for that and all their help along the way. And so I want to talk about funding and research, right? I wouldn't be doing this if I had zero money. So obviously, Oregon State was able to give me money through teaching and allow me to eat food and survive. And then the U.S. Forest Service provided funding for the research. So no matter what I needed to get the research done, or and then GRAs over the summer, so I didn't have to worry about teaching and still was able to make money. It was incredibly helpful for the process. And then I want to give a thank you to the IB community. this is, I think, the biggest strong suit, as Bob had mentioned earlier, the biggest strong suit of our department here at the university. I've had friends be grad students here and talk about how they don't even know their cohort or really the people in their lab, and there's not a lot of community there. But from coffee hour to my first time snowboarding to like family dinners to hikes, all these things, which one, I never thought I'd snowboard in my life, and then I ended up being on, it's crazy. But it is insane the support we have for each other. I think a lot of us would have a much harder time if we all felt isolated in this. And like, if you turn around and look at the room, we got a decent amount of people in here. It was a little scary when I first started, everyone walked in. But the insane support we all have for each other is amazing, and one of my favorite things about being in this department. And with that, there's so many more people, right? If I was to give a slide to everybody who deserved it, this would just be a thank you letter and there'd be no research being talked about, right? So, but I wanted to make sure a lot of people got their name on here. And note on the bottom right, anyone who goes to or has gone to coffee hour is also on this list, because that's a lot of people, and coffee hour is very important to me. I think another part of community building. And then I want to get into the personal acknowledgments. We'll see how my emotions handle for this. Yeah, we'll see. So my family, the first thing I want to say is, like, statistically, as Bob mentioned, we're not from Bel Air. Statistically, I shouldn't be in this position. Like, there's no reason for me to be up here on stage at an R1 university giving a master's defense talk, right? But it's through the insane support my family gives to each other, not just me, but like the way we all support each other and love each other is something I like. It's unimaginable. And if you've been around my family, you kind of understand. But it's unimaginable, and I appreciate them all deeply for that. And this is where things might get hard. Let me take a sip of water. Nicole. Okay, this is gonna be rough. Okay, so me and Nicole have been a partner for nearly 10 years now, and she's like the primary reason I started going back to school in the first place. We were working together at Office Depot and we're having conversations, and she just asked casually, "Oh yeah, so like, where do you see yourself in 5 years?" Which at the time I was like, "That's a good point." Like, I wanted to go to school, but I didn't know what I'd be doing in 5 years. And it was like 2 days after maybe that I was like, "Okay, go to Chemeketa. We're going to community college. And we're getting all started." And even outside of that, through like my insane levels of stress, when I'm having great times and we're having fun in Costa Rica or whatever it may be, she's been always there and always supporting everything I do. And thank you. She's crying, is going to get me. We're good. All right. And then I'm excited for our next chapter. Don't get me. Thank you.
Okay, so just if this is the first event you've ever been in, we never really do this, say it enough. So now is the time for the public to ask, you all are the public, to ask questions of Dustin. And I would just ask that his committee members reserve their questions for the little tiny exam.
Okay, tiny.
All right.
Yeah.
Call them.
So, excellent work. Well done.
Thank you.
Quick question about the geographical range.
Yeah, do we go back to it?
Really understudied.
Yes.
How much do you trust the geographic range? Do you think it's
Yeah, so actually, I do want to go back to it because that's a good point. Because there is actually a lot there to talk about. It's a lot farther back than I thought. We're almost there. We'll get there very quickly. Really, I think like I trust obviously what is there. We're almost there. There's a lot of slides. If you haven't picked up, I talk really fast, so I kind of need. No, that's not the range one. I talk really fast, so I need a lot of slides to kind of parse me through this. Okay, right there. Stop moving. Okay. Yeah. So I'm pretty confident in this stuff, right? This here is pretty good, solid, right? We know like the environment they need and all that. What I think there's room to like really look at is this up here, right? This location right here was the type locality. That's where they were discovered, right there in Washington. And there's a lot of suitable habitat in this range, right? And they even make it up here in Vancouver Island, which like may not be the most optimal for an ectotherm, right? They're making it work. So I think in here, I trust this portion of the range, right? Because when we start getting here, we're getting into Cascades and east of the Cascades, where you're getting a very different climate that might not be as suitable for them. But I think here, there definitely warrants some search to kind of expand that range. Yeah.
Since we're on this slide,
Yes.
dear God, do not change it.
Yeah, I know. I figured that out.
The down in like Bishop, like
Yeah.
the lowest part of
Yeah.
the range.
Yeah.
Do you feel like now that you've looked at Vancouver and now Oregon, for those growth rates, do you think you have predictive abilities to say that the lower down you go, there might be a different growth rate?
I'm glad you asked that because there's a part I could have put in here, but I was like, I think a question will come up. So I wanted to be a question. So I worked with David Hubert and we did a little bit of preliminary data that I would have wanted to include, but we were trying to get this wrapped up, so it wasn't time to do a whole new analysis. But even in those two groupings of the population we have here in Oregon, we were finding differences in their size. So I think that if you were to reach this all the way down, I think we'd find differences in that based on our preliminary study, just in order.
You mentioned at one point that they prefer oak savannas, or
Yeah.
that's something they like.
Yeah.
But in the last 50 years, as we know, oak savannas have
Yeah. Yeah.
in Oregon. Do you think this has reduced their rate, played a factor in their growth rate?
I mean, it probably plays a factor to some extent, right? When habitat destruction is never really great, but also with them seeming to be fine with relative levels of human disturbance, right? Being next to that farmland, I think they're probably making do with these changes.
Really interesting talk. My question, I'm no snake expert.
Yeah, I guess I am.
Looking at your ranges where you're seeing faster growth rates, the southern range is slow
Mhm.
growth rate, the north range. Do you see that correlate with other snake species that have this fast range?
Yeah, and sometimes so there's a study that was done on garter snakes and looking at elevation, right? We have also different climate. Sometimes it depends on the snake and how they handle it, and then also a lot of it can be prey availability, right? So if you have nice abundance of prey, you're probably going to be doing better and thus be able to grow faster and do all these things. So that could be one thing, but it is common at these colder climates where you have less temperature that you're going to have a lower growth rate.
Thank you.
So your shot features disabled on Zoom, so.
Oh, okay.
So you just went on this huge journey academically.
Yes.
What was your favorite part? What was your takeaway?
Okay. I takeaway. We'll start with the takeaway. I think it's that there's this whole group of snakes. I'm assuming there's a lot of people in here who probably didn't know what a sharp tail was before you read the title of my talk. Is that there's a lot of these animals around that are in some areas easy to study or relatively easy, but not looked at really at all. And I think the takeaway is that we should put more effort into these small understudied animals in the area because they could have huge ecological impacts, right? If we don't know anything about them, we don't know their impact. So I think that was my big takeaway. My favorite part, I think the favorite part is just going out and holding the snakes. They're really fun. And their sharp tail's really fun because they're poking around while they're climbing around you. It's really fun. So I think the best part is going out and handling the snakes. And then also running data is also kind of fun when you get a cool graph, making that graph, the seasonality and thing. I like how it looked, right? The peaks line up with the cross of the weather. So I think sometimes messing with the data was a good time. Yeah. Yeah.
Okay, so I was gonna say Eric's question is a way more wholesome final question than this one, but then you gave me the segue to the data analysis because you show the seasonality and even have a little bit of a story. We'll call it a hypothesis there,
Yes.
maybe how it's connected to nocturnality,
Yeah.
slug behavior, and so
Yeah.
cool hypothesis. How do you know? I think your y-axis was just total observations, right?
Yes.
How confident are you that there isn't a sampling bias issue at play there, and how could you account for it?
Well, I think for sure the sample bias would be the daytime sampling, right? I think
Yes.
that was the point is that the future studies should include nocturnal efforts,
Okay.
right? Because most of the studies were, most of the sampling efforts were done in the day. And so we missed that entire factor of, and that's why they're talked about as being not really surface active. But of the evidence I've seen or heard from people is that in the summer, they seem to be surface active, right? Not necessarily under, but on top of things searching, which would change what we know about the snake as a whole, right? It's not always hidden underground, that in the summer months, it's on the surface, right? So there's definitely a sampling bias, and which is why I think.
What about total person hours out in the field? Is that pretty consistent through the years?
Through the years, through the years, it goes up and down. So there's groups of years where there was a lot more efforts.
And do those correspond to your peaks of activity? Different seasons. Was that when Richard took his yearly trip to the true Mediterranean in the middle of the summer, right? Was that pretty consistent through the years, the total amount of effort, or could that be biasing the peaks and the seasonality that you're seeing?
Yeah, so the first season I was out in those early years had more effort than some of the future years.
Okay.
But across the board, we saw the same trend in season activity. And even when I had first started, I went out and obviously very small scale, one season, I saw the same trend very quickly in while going out and searching for them.
Was going off of that question
Okay.
kind of, but because of how many years worth of data you have, do you
Yes.
see changes, subtle changes of activity over time based on variation in temperature and precipitation?
No, I do think, especially as we get into the later parts, I think if there variation is going to be much more in these later the 2020 years, we're seeing more efforts that are selected for certain times, right? Which will affect the rate at which there's seasonality. But I wasn't seeing any change in seasonality as it gone. If that's what you were asking,
Yeah.
I think it was cool. You already hand up a couple times.
Finally, be quick.
You did say everyone else can go first.
So I'm really interested in not only like obviously I'd love to go look for some truck tails in some weird new places. But what's interesting to me is that we talked about their diet being slugs, but that they're probably not eating banana slugs, or at least not as adults, and that they're heavily tied to these non-native
Yeah.
slugs
Yeah.
that were introduced,
The first sighting was 1933.
right? Well,
Yeah.
shark tails were around
Before that.
long before
Yes,
that.
yes.
So what are they eating in a more natural habitat, or at least in a more, further away from potential invasive slugs? And how can we find out what they're eating, and how could we maybe use that to infer maybe some more of what's going on in the ranges that we suspect they might be, but they haven't been found?
Yeah, I think before the invasives got here, I think the nice part about a lot of the invasives for the shark tails is that they're smaller bodied than a lot of our native slugs, right? The banana slugs being the big one. So there's more time in that animal's life that it can get preyed upon by a shark tail, right? So before, my guess is just either slug eggs and small early life slugs, right? And then that's why the diet analysis could if we're seeing that they're eating worms or salamanders or whatever it may be, right, can help fill that question in. But and then what was your second part of your question?
Oh, just how can we use that information to better infer maybe what's going on in these ranges that we suspect they might be, or maybe more broadly, like what did you take away from this study about how we can study difficult to find, in this case snakes, but
Yeah.
organisms?
Yeah.
You know.
Well, so I guess the second part I have a very quick answer for, which is adjusting your methods to fit the animal, right? When we're using the same typical methods that people always use with normal artificial cover objects, and adjusting that to fit the animal and what might suit it, and not being afraid to experiment with new things like a gravel pile could be a way to sample for these hard to study things. And as far as potentially what they're eating before influencing their range, I guess it really would be looking at these things that they're associated with or potentially preying upon, right? If we got a diet analysis and looking where they exist in these disjunct parts of the range or right around the range, and then trying to see if they also exist in that area.
Cool.
Yeah.
It seems like some of the populations might be fairly isolated. Maybe you guys that. But if that is the case, do you think that might be contributing to some of the variation you see, especially around age of social maturity?
In our populations?
Yeah, just compared to Vancouver.
Yeah. I think the Vancouver isolation part is the good example in the variance, right? Because not only is it more northern, right? More northern latitudes are typically colder. And then it kind of has this unique climate because the influence of the ocean around it. And so that change in climate, which can have different rainfall, different temperature pattern, all this kind of stuff, I think is a big part of playing into the growth. And then also, I don't know how much we know about the prey availability, abundance, and nutrition levels in that area.
So unrelated to other question, but regarding hatchling morphology that you mentioned,
Yeah.
using a non-stressful way to measure if it's a male or female. Given so there's this little difference between the different tail lengths between sexes. Are you able to estimate or pinpoint the sex based on a certain length range of the tail?
Yeah. So it's the total length of the tail is longer in males, and then because of that, the ratio of body sizes from the tail to the body size is longer.
And there's a specific ratio that corresponds.
Yeah. I forget these. I have the number. It's 13. It's 13.4% of the total length compared to 11, I think, and it's a very small variance range. So there's not a lot. There's no overlap there.
Do you think this is transferable to other species, and
it's worth looking at for other species, right? And I've talked to Mark about it, that in rubber boas, when they're born, no different than tail length, right? They're the same. And it gets different as they get older, but there's no difference at that hatching point. So I think it's worth looking at and trying to find these non-invasive ways to sex snakes. I know with some, it's the tip of the face to the mouth gate can be an indicator for sex. So it's the relative tail length is not it for every snake, but I think it's worth looking into these non-invasive and then thus easier, more accessible sexing strategies, right? In all honesty, that little one on my finger, I definitely could not sex that, right? With the old school means of a probe or even the hemipenes, right? Without the real risk of damaging it. Yeah.
So since we're at the end, I was just gonna say, really, I should jump up. So if it's meaningful to you folks, so I've been working on snakes for decades, and I handle literally tens of thousands, or over 100,000. I don't know. And for whatever reason, as Dustin told you, the gold standard for snakes is you measure snout bend length. You've got a snout, you're probably alive. You may be missing half your tail, so you can get predators chew your tail. So people were using snout bend length, but they were not reporting tail length. And so it's only really within the last, I don't know, I'd say 15 years or so that there was a plea that came out from our tribe and said, please, by all we please do total length and tail length. And so this may seem just blatantly obvious to folks, but it just was not what people did. And it's because tails can be missing, so you don't say this, you can report the total length, but it's, that's a damaged tail. So this kind of thing, there's a lot of data that be looked back into and look at population demography that just has never been done. So it sounds very subtle, but it's actually a fairly important finding. Okay, with that, let's thank Dustin.